We're going to talk about the three different structural types of capillaries that we have. We know that capillaries are pretty thin, and this is where exchange is going to take place between the contents of our blood and the contents basically outside of our blood. The thing about capillaries, though, is that not all capillaries are going to have the same level of permeability. So we have some arteries that are very highly permeable, allowing large cells to pass through them. And we have capillaries that are not that permeable, and they're only going to allow things like fluids and small solutes to be able to pass through them. So the first type of capillary we're going to talk about is the continuous capillary, and this is the least permeable type we have. Then comes fenestrated capillaries, and fenestrated are going to be a bit more permeable. And then the most permeable type of capillary we have are known as our sinusoids. So let's start with the continuous capillary right here. The continuous capillary, as I said, this is the least permeable type of capillary we have. The continuous capillaries are usually abundant in our skin and in our muscles. They're not that permeable. They're only going to allow things like fluids and small solutes to be able to pass through them. Um, the continuous capillary, it's only made up of the tunica intima. Actually, all of your capillaries will only be made out of the tunica intima. You can see right here the endothelial cells, and the endothelial cells are connected by some tight junctions, which are basically proteins that are holding the cells together. But you notice how that there are some gaps right here known as the intercellular clefts, and those gaps are going to allow very small things like ions, like potassium, sodium, and fluids to be able to pass out. To actually put more restriction on those continuous capillaries, there is that basement membrane layer, which kind of puts restriction, preventing large things from passing out of that continuous capillary. And to add on to the restriction, there are those cells right here known as the pericytes. And those pericytes are like smooth muscle-like cells, and they basically add on to the restriction, preventing large things from passing through those continuous capillaries. Capillaries are very narrow to the point that only one red blood cell can pass in a single file. So you're not going to have two blood cells in pairs passing through a capillary. That's how narrow it is. Only one cell at a time in a single file. Basically, they're going to move in a single file through that capillary. Fenestrated capillaries, the word fenestrated actually means pores. And the reason why we call them fenestrated, because take a look right here at their endothelial layer, and you would notice that there are a bunch of pores within that endothelial layer. Fenestrated capillaries, because they have those pores, they're going to be more permeable, and you would find them in areas where where you have filtration taking place, such as the kidneys. Because if you think about it, the kidneys filter out your blood, and so you want to get rid of a bunch of waste. And so you would want to have a bit more permeability to have an easier time getting rid of that waste. You would also find fenestrated capillaries in areas of absorption, so therefore you would find them in your small intestines, because this is where you digest your food, and so you would want to have an easier time absorbing those nutrients. So you don't want the capillaries to have a lot of restrictions on them, and so it would be a better idea to have those fenestrated capillaries in these regions. Let's take a look at this type of capillary, and those are the most permeable type of capillary we have. You can see that the pores, those fenestrations, are way larger. Even the intercellular clefts, notice how it's pretty huge gaps. And so that means that those capillaries can actually allow pretty large things to pass through, meaning cells. So red blood, like not red blood cells, usually it's white blood cells, the ones that are going to help you fight off infections. They're actually going to be able to exit out completely out of those sinusoid capillaries. You would find them in areas like your liver, your bone marrow, your spleen, even the basement membrane. Within those sinusoid capillaries, you can see that it's broken up, so it's not going to put as much restriction on those sinusoid capillaries compared to the other capillaries that we've talked about previously. If we take a look at this image right here, it shows that direct contact between your arterioles and your venules, and the blood passing through from the artery side to the venule side through that capillary network, or we refer to it as the capillary bed. This is the arteriole, and we refer to it as the terminal arteriole. So it's the terminal, kind of like it's the last one before you hit that capillary bed. Those capillaries right here, we refer to them as the true capillaries, and this is going to be a venule. And if we want to be more precise with the name, we call it the post-capillary because it's after that capillary network, so the post-capillary venule. Now, those precapillary sphincters right here, those are made out of smooth muscles. And so when these smooth muscles are relaxed, those sphincters open up. What happens when those sphincters open up, when the blood is rushing, into that terminal arteriole, and those sphincters open up, the blood is actually going to make its way into those true capillaries. And when the blood starts flooding this region right here, those true capillaries are usually found on top of cells, on top of organs. And as we said, capillaries are pretty thin and they're pretty porous. Gas exchange, nutrient exchange is going to start taking place between those true capillaries and between the organs or the cells that you have the true capillaries right on top of. And then, as you can see, the blood is going to start making its way into the venules. Now, what happens when those precapillary sphincters contract? They're basically going to close. And when they close, you would notice that the blood is not going to enter and it's not going to make its way into those true capillaries. It's just going to go from the terminal arteriole into this branch right here known as the meta-arteriole. And from there, it's going to enter into the through-fare channel and then drain into the post-capillary venule. When you don't have any blood entering into those true capillaries, you're going to skip that exchange. You're not going to have exchange between your capillaries and between your tissues. What's an example of when those sphincters would open and close? Let's say that you just ate And so the food makes its way into your stomach, and from there into your small intestines, where you're going to start digesting the food that you just ate. Now, after digesting the food, you would want to absorb the nutrients. So therefore, those sphincters are going to open up so that you can actually start absorbing the nutrients from the blood, basically after you've eaten a meal. But if it's a, you've eaten a meal and you've already digested your food, and so and you haven't eaten anything else, then the sphincters are actually going to close, because right now you're not, like, digesting food, and so therefore there is no need for any exchange to take place, and so the sphincters are going to close up, and the blood is just going to directly make its way into those post-capillary venules. One thing to note about those post-capillary venules is that they are a pretty unique type of venules, and the reason why they're pretty unique is that they act as a capillary. They are pretty thin as a capillary. They only have that tunica intima, and so therefore actually white blood cells can pass out that post-capillary venule. Any other venule, by the way, is not going to act as a capillary. That's kind of like the only, or that's like an exception that we have. Other venules are still going to have all three tunica layers, and they're not going to allow any exchange to take place within them because they're going to be much thicker. Now, we've already talked about how veins, their lumen are much larger compared to arteries, and we've talked about how veins, the blood pressure in the veins are much lower compared to the blood pressure in the arteries. Actually, veins act as our blood reservoir. They can actually hold up to 65% of our blood supply. Especially the veins in your lower limbs, they're going to have to push the blood towards your heart. Remember, all veins push the blood towards the heart. And so to help out, prevent the blood backflow, we have those valves within our veins. So let's go ahead and take a look at this image right here. Here are the valves. Not all of your veins, by the way, are going to have those valves. Usually the veins that have those valves are the veins in your lower limbs. Because they're like, the blood will have to move such a pretty long distance to make its way to the heart. The veins in your abdomen or in your thoracic region are not going to have those valves. And definitely, by the way, arteries are not going to have those valves either, because the blood pressure in your arteries are much higher compared to the blood pressure in your veins. And so to prevent the blood backflow, we have those veins right here. So as the blood is being pushed upwards, once the blood bypasses the valve is going to shut close to prevent the blood from going backwards. What also helps push the blood towards the heart is the skeletal muscles in your legs. When the skeletal muscles in your legs contract, that's actually going to help squeeze those veins, helping push the blood upwards. When individuals have problems with their valves and their valves are becoming leaky and they're not functioning properly, the blood backflows, and that's when you start getting those varicose veins in your legs.